I can hardly describe how delighted I am to be here, because last time, and I don't even remember whether it was in this room, it's at least 25, 30 years back. And at the time, I talked about my former life, and that was phage mu and recombination. So it will be a very great pleasure for me, and also a great honor today to tell you where we stand in terms of analyzing our favorite system, how a fungus colonizes a plant. And in this case, it's a biotrophic fungus. So one that establishes a very intimate relationship with its host, has no intentions to kill the host. Quite the contrary, the host has to stay alive. Okay, so let me start by basically introducing the system, and I would like to do that in a bit more broader sense by showing you this group of fungi, Smut fungi. And I've picked this picture for a particular reason. So what you see here is Ustilago maydis on corn, Ustilago hordeae on barley, Ustilago bromivora on brachypodium, and Sporogenesis morellianum also on corn. So all these fungi cause diseases. These diseases are not very relevant in agriculture, which may be a problem if you work with this fungus in the US. In Germany, there is no problem. You don't have to justify. You can simply convince people that I'm excited about the science, and this is why please give me money, okay? So I'm showing this picture for one particular reason, and that is, you see all these symptoms develop in the flowers. And with one exception, this is what is happening. So it's only that you get this black masses of spores produced in the flowers. So if you work with such a system, pretty bad for you, because you have to wait. So in case of maize, if you work with Sporogenesis, this means 3 months. And if you have worked with phage before, this is just not acceptable. So I decided on this one, which is also infecting the flowers, but it also infects small plants. And these plants were about 6 days old when they were infected, and you see these tumor-like structures develop on all above ground parts of the plant, and it takes about 6 to 8 days to see these symptoms. So this is a system which I think is manageable. And it also means you don't need a lot of space in the greenhouse. So this fungus is Ustilago maydis. Let me briefly introduce it. It has a rather complex life cycle. So we start out with the spores, which they are diploid, they germinate, produce haploid cells. And these grow like yeast by budding. However, if you have a pure culture of a haploid, it's not infectious at all, no symptoms whatsoever. So in order to get to the pathogenic stage, you have to have 2 cells which differ in mating type, and they will recognize each other with the help of a pheromone receptor system. They will grow towards each other, fuse, and then there is a very dramatic morphological transition, and you get a true hypha. However, this hypha looks odd because all the cytoplasm from the original mating cells moves to the tip, and this is where growth occurs, and the rest is basically vacuolated, and it's empty. And it's separated from the growing tip by regular space septa. So it's this dikaryon which is infectious. And our initial work really dealt with the question, why do you have to have the dikaryon? Why is only the dikaryon the infectious form? And we found out that, yes, indeed, it's regulated by the mating type loci. So you need mating. And then in the dikaryon, it's a homeodomain complex of 2 proteins that needs to form. And this is then the active regulatory component for all subsequent steps. This is at least what we thought at the time. You can also visualize this and get rid of the dikaryon by making a haploid strain that just expresses this regulatory complex which is active, and then a haploid strain is pathogenic. And this has really opened up forward genetic approaches, and basically a lot of the work we are currently doing would not be possible without such strains. Okay, nevertheless, in nature, it's this dikaryotic stage, which then, upon contact with the leaf surface, where hydrophobicity and hydroxy fatty acids are sensed, forms an aprosorium, which is the infective structure, and this then penetrates directly. So what you see here is that this fungal hyphae is inside the epidermal cell, but nevertheless, it's not in direct contact with this cell, because the plasma membrane of the host cell, upon penetration, has invaginated and really surrounds the hyphae like a very tight glove. Later on, you get massive proliferation and branching of hyphae in the apoplast, and at some point, nuclear fusion and spore formation. I will come back to that in the second part of my talk. So I want to show you how this looks in real life. Here we have the fungal hyphae in red penetrating, and the plant actually expresses the plasma membrane marker fused to YFP. And you see at the point of penetration, this plasma membrane invaginates and surrounds the hyphae. And here you see this hyphae in the epidermal cell, and you see it's completely and tightly encased by the host plasma membrane. So there are no specialized feeding structures in this system, but you have this very extended interface between fungal hyphae and plant plasma membrane. And this is where all the action takes place. At later stages, you find the fungus close and inside the veins, and this is presumably where the fungus gets nutrients for the massive proliferation. So to actually get there and enter this tissue is crucial. And then again later, you find huge fungal aggregates, and it's in these aggregates that spore formation, that nuclear fusion and spore formation is initiated. So Ustilago is a biotroph. Biotroph means the plant cells, everything surrounding these fungal hyphae stays perfectly alive up to the very late stages when you have spores, and then at some point these tumors that develop rupture, dry up and rupture, and then release the huge amounts of spores. So what we have learned over the past 10 years or so is that the interaction with the host is governed by secreted effector proteins. And we were, at the time, incredibly lucky to come across these proteins. We did not fish the respective genes by forward genetics. We now know why we didn't manage. But we came across these genes when we had the genome sequence. So it's about 300 genes, and they code for conventionally secreted proteins. So they all have a signal peptide at the N-terminus. And this is pretty much the only thing you can recognize, because the vast majority of these effectors lacks an annotated function. So you cannot predict anything. However, more than 90% are found in all Smut species. So at least the relatives have the same genes. And these genes are very poorly conserved, and you can actually detect them in the genome easily because most of them are poorly conserved. The reason why we were so successful in picking up these genes from the genome sequence was that it turns out that about 25% of them, of the respective genes, are arranged in clusters in the genome. And I should also emphasize that the expression of the effector genes is linked to the phase inside the plant. So some of these effector genes are already upregulated when the fungus is in contact, and others are expressed at later stages. So we classify the effectors, as in other systems, into apoplastic effectors, that is, proteins that after secretion end up in the space between fungal hyphae and plant plasma membrane. And we distinguish these from cytoplasmic effectors. These are proteins which also end up here initially after being secreted, but then they are taken up by plant cells, can actually spread via plasmodesmata to neighboring cells, and they have their function inside the plant cell. So we have about 300 effectors, unknown proteins, and we would love to understand what they are doing. And this is a true and real struggle to work on something where you cannot predict a single function. And the result is that despite 10 years of work, we know the function of 4 effectors. And this is not only my own group, okay? So the group of Gunther Döhlemann has shown that one of these, has characterized 2 of these apoplastic effectors, PEP1, which is an inhibitor of a peroxidase in maize, and PIT2 inhibits a group of cysteine proteases. And my own group has characterized 2 of these cytoplasmic effectors, a chorusmate mutase, which is taken up and changes assay levels, lowers salicylic acid levels in the infected tissue, which is beneficial for the pathogen, and an effector which induces anthocyanin. And I briefly want to talk about this effector so that you know what it takes to analyze the function of one. This is published, and I promise I will then come to unpublished work. So this effector is a virulence effector. If you delete the gene, you can see, compared to the wild type, the tumors are smaller. They are still there, but they are definitely smaller. But most prominently, this red pigment is completely absent. And if you complement, you restore tumor formation as well as anthocyanin formation. And this is the effector which we call TIN2. What you can also visualize is that TIN2 mutants can colonize plants at the early stages, really to levels comparable to wild type. But if you look at later time points, there is a dramatic difference. So it's really late proliferation, which is attenuated when this effector is missing. We use comparative genomics to actually pick important sequences in these effectors, important domains. And here you see an alignment of TIN2 proteins from different Smut fungi. And you see there is very little conservation except for the C-terminal domain. And it turns out if you just delete a small 5 amino acids from the C-terminus, the protein is no longer functional. So how does one find out where this protein functions? So in this case, we bombarded maize tissue with a construct expressing TIN2 protein without its secretion signal. So it should be expressed inside plant cells. And what you can clearly see is if you do that, you get anthocyanin production. And if you do the same experiment with a protein lacking the C-terminal 5 amino acids, there is no anthocyanin induction. This illustrates that this protein is functioning inside plant cells, so it must be translocated. I should also say that this experiment worked only if we take maize plants for the bombardment, which we had pre-infected with a TIN2 mutant. So obviously the infecting fungus changes the metabolism in such a way that you make enough precursor for the biosynthesis of anthocyanin. So with a lot of work I don't want to go into, we could then visualize that TIN2 protein actually stabilizes a maize kinase, and this kinase is responsible, probably in connection with this R transcription factor, for the massive upregulation of anthocyanin biosynthesis. So if we have a fungus that lacks TIN2, this protein, this kinase is degraded. And what we see instead is that there is an upregulation of lignification. And lignin and anthocyanin use the same precursor, p-coumarate. And we believe that the fungus channels the pathway into anthocyanin biosynthesis to prevent lignification. And as a result, you see that at the late stages of the infection, where the fungus, and you see that here with these small circular structures, where the fungus is in the veins, there is very little lignification. However, if you do the analysis with the TIN2 mutant, this tissue is heavily lignified, and you do not find the fungus in there. So this is what we think this effector is doing. To figure this out, this did cost one postdoc about 3.5 years. Okay, so you can imagine what we are up to if you want to understand all 300 effectors. I'm not going to talk about effector function. There's a lot going on, but we don't have complete stories. However, I want to address a really enigmatic issue which has kept us working for almost a decade. And that is how eukaryotic pathogen effectors are actually translocated from the pathogen to the host, and which effectors are translocated. So if we look at the 300 effectors which are produced by Ustilago maydis, they do not have anything in common, except, I mean, there are some gene families, yeah, but otherwise you cannot predict. So for example, with oomycetes, we have crinklers and we have RXLR effectors that have conserved features. We do not have this. So just from bioinformatics, we have not been able to classify the effectors and say these ones are apoplastic and these ones are cytoplasmic effectors. So I will now try to convince you that we have an assay that allows us to do that. And the result of this assay came and is quite a surprise. Okay, like everybody else, if you want to know where your protein goes, you make a fusion. In this case, we fused the TIN2 effector, which we know is translocated, to mCherry, and in addition, we hooked it up to a nuclear localization sequence, thinking that this would then concentrate all the transferred effectors in the nucleus. That this is working, and this construct is working, is shown here, where you see that if you transiently bombard maize cells, you find this fusion protein exclusively in the nucleus. However, if we now do the real experiment and express this fusion protein by the fungus, we do find staining of the fungal hyphae inside the plant. And because the effector is secreted and the hyphae is encased by the plant plasma membrane, the effector cannot fuse away, and this is the typical distribution which we see for a secreted protein. However, if you now look at the nucleus of the plant, there is no staining whatsoever. And in this case, for the infection, we used silk of maize because there is basically no background fluorescence. And still, we are unable to see it. So we have never been able to see a fusion protein to a fluorescent protein translocated to the plant cell. I don't want to go through this complicated slide, just to tell you we have done plenty, we have established plenty of other assays where we used a split GFP system for effector translocation, where we modified, got transgenic maize plants, where effector translocation would excise this stuffer by flip recombinase, and then you would get Gus expression. None of this worked. Very disappointing. I'm happy to discuss why these assays may not have worked. Because now we think we have an assay which is working. And this assay is based on biotinylation. So the biotin ligase reaction works in such a way that you basically have a lysine residue, and the biotin ligase will ligate this to biotin. And in this case, we use the biotin ligase of E. coli, BRA. It turns out a lot of work has been done on these biotin ligases, and it has the substrate that is recognized has been thoroughly analyzed. And it's actually a very short tag, a 15 base pair sequence, and you see here the lysine residue. It's a so-called biotag. So if you biotag the protein you're interested in with this tag and then express BRA, you should get specific biotinylation, which you then can document. And this has been established in yeast, in mammalian cells, in insect and in plant cells. So we thought we should have a good chance to develop this into an assay system for effector translocation. So the idea is very simple. You have a set of apoplastic and cytoplasmic effectors. You fuse them all to this little biotag, let them secrete by the fungus, so in the homologous system. And then you have a plant which expresses the E. coli biotin ligase. And if the effector is translocated to the plant, it should be biotinylated. If the effector stays here in the apoplast, it should not get in contact with the plant. So that was the idea. Well, if you have ever wanted a transgenic maize plant, you know this takes time. So we had to wait, and we had to do something in between to convince ourselves that this assay may be working. So the idea would then be you transfer your proteins, your effectors, biotin, the AVI-tagged effectors to the plant, and then make a plant lysate, do an IP with streptavidin magnetic beads, and then you detect your bound effectors via an HA Western blot, and the effectors also have a short HA tag. So to convince ourselves that this is working, we actually expressed several versions of the effector, initially in Nicotiana benthamiana, and we co-expressed or left out expression of the biotin ligase. This just shows that in all cases we got expression of the effector. So if something doesn't work, it's not because the effector is not there. So in this case, we expressed the effector with its own secretion signal in the presence of BRA. And you can see basically then nothing, because the effector is eliminated from the cytosol, it's secreted and should not get in contact with BRA, should not be biotinylated. In this case, we express the biotagged effector without the biotin ligase, nothing. In this case, it's the secreted effector without its secretion signal, biotagged, and BRA, and you get a massive signal. And in this case, it's an effector without the biotag and BRA, and there is nothing. So we were extremely happy because this shows that the system could work in principle. Then we had to wait for more than a year and got our transgenic plants. We decided to try a number of effectors. So the two translocated effectors, CMU1 and TIN2, the two apoplastic effectors, and an effector actually from the flax rust fungus, which is translocated in that system, AVRM, to see whether the system would also work for heterologous effector. In addition, we made sure that we have good controls. And we thought the best controls we can do is biotag secreted mCherry. This should definitely stay in the apoplast. And SUC2, which is an invertase, which should be secreted but remain associated with the fungal cell wall. So this should definitely also not get in contact with the biotin ligase. So I mean, just I don't think I have to run you through, but we basically do an IP and split up the IP so that on the one hand we verify that the effector is actually made and the quantity, and then we do an enrichment for the biotagged protein. And it's working. It's working. So, for example, we always have plants that express the biotin ligase, and we have plants that do not express. And we infect plants which lack the biotin ligase, and you don't see any biotinylation. However, if we take plants that have the biotin ligase and express them with infect them with the fungus, secreting the biotagged TIN2, you get a biotagged version that is a transferred version. And this also works with the course-mate mutase, and it works with the unrelated AVRM effector from rusts. So we were incredibly happy. Then you do controls. And controls, as you all know, do not always work. So unfortunately, not always, but sometimes we also got invertase biotinylated, and even more or as irritating, sometimes we got cherry biotinylated. It was a very, very bad time in the lab, okay? So, and I mean, and hopefully the right explanation was when you break open these cells, these infected maize plants, I mean, it takes time, at least if you do a lot at the same time. Maybe this is a problem that is occurring during a plant cell lysis. So how does one cope with the problem? Well, we looked in the literature and found out that you can inhibit biotin ligase activity by raising the amounts of sodium chloride and by adding glycerol. And in addition, we decided to modify the system in such a way that rather than having a separate control and a separate effector we wanted to test for transfer, to actually express both AV-tagged proteins from the same fungal cell. So in this case, it was the invertase was tagged and mCherry was tagged. And you can see this addition brought a major improvement for the invertase. However, mCherry was still biotagged. And in this case, so it was a strain expressing the corsmate mutase and mCherry, and we find both proteins biotagged. So we always find mCherry biotagged. What does this mean? We decided we better verify our data and find somebody who does EM. And lo and behold, we find mCherry biotagged HA inside the plant cells. And if we do the same analysis with biotagged SUC2, we find this associated with the fungal hyphae, but we do not find this in the plant cell. So we believe the uptake of mCherry is real. So let me summarize this part of my talk. So we believe we have established an assay that relies on and the physiological delivery of effectors by Ustilago maydis, and that we are actually able to do this in the homologous system, I think is very attractive. The assay is highly sensitive. We have now been able to detect uptake of all known cytoplasmic effectors, but I have to admit there is only 2 that are known. However, we have also seen that some of the apoplastic effectors are taken. And the flax rust effector AVRM, which is heterologously secreted in this case by Ustilago maydis, is also taken up. And I hope I have convinced you that this fungal invertase, rather than mCherry, may be a really good control to be included in this assay. So if I sum these results up, what does this mean? It basically means there is no specific uptake mechanism for Ustilago maydis effectors. Anything goes, except something is holding its back. And so I think rather than asking what determines uptake, we have to ask ourselves what determines that it's not taken up. And right now, except for saying if it's attached to the fungal cell wall, it's not taken up. I cannot tell you this, but we will look into this. So we are pretty excited because this assay can now be extended to actually ask how does uptake operate. Okay. I want to shift gears and move to another story which also came quite as a surprise. And it's a story which tells you, in the end, how little we know. So the expression of effector genes is linked to the biotrophic phase. If you grow the fungus in culture, there is no expression. So a while ago, we were intrigued by work from the Mathieu Rep lab in Amsterdam on Fusarium, where they identified a warp box protein, SGE1, as a regulator for several of the effectors in this fungus. And it was also clear from their presentation that Ustilago maydis had two such warp proteins. So we wanted to know whether any of these regulators, transcription factors, might be involved in effector gene regulation. One gene had no phenotype when deleted, but the second one, which we term ROS1, has a very intriguing phenotype when deleted. So first of all, the gene encoding ROS1 is differentially regulated, and the gene is actually induced, begins to be induced at 4 days after infection, and is then strongly upregulated in the infected tissue and stays on until late. So a normal infection, and here we start at day 4, basically looks like this. So this is wild type. The cells begin to form these aggregates, then make these huge structures, which are actually in which the fungal hyphae are connected via some matrix, and it's in these structures that you can then get the transition to spores. So after 12 days, the cycle is complete. If you do the same analysis for a ROS1 mutant, you basically see that everything is fine until day 4, but then nothing changes anymore. And there are absolutely no spores produced. So to learn at what stage exactly it is that this mutant starts to have problems, we had a closer look. So at these sporulation stages, or prior to sporulation, you get nuclear fusion. And the spores that come out are diploid. And so we have here looked at the wild type fungus, and at this stage, I think this is about day 6, you can see that per cell we have only one nucleus. So obviously the nuclei have fused already. In the mutant, however, you can clearly see there's always 2 nuclei. So they are unable to fuse, to undergo karyogamy. Presumably associated with this is that fungal biomass does not really accumulate in the mutant. So in the wild type, there's a massive proliferation, which we can now link to the diploid hyphae, and this is basically absent in the ROS1 mutant. What is also completely missing is the polysaccharide, for which we do not know the nature, but you can stain it with basic fuchsin, and you can see it gets this red, gives you this red coloring. This is completely absent in the ROS mutant infected tissue. So we asked what are the genes which are differentially regulated, and we actually did 2 things. One is we compared wild type and mutant infected tissue at about 6 days after infection. And the other thing we did was we can induce fungal hyphae artificially with a genetic trick, which I don't want to go into. And so you can induce the transition from budding growth to hyphal growth. However, if you co-express in these strains ROS1, you block this completely, and instead these hyphae get really fat and start to make septa. and these septa have individual nuclei. So you induce mitotic divisions. Okay, and so we did arrays after inducing these morphological changes, and then we combined everything. And this was really amazing, because it turns out that ROS1 is a regulator, and it's actually down-regulating more than 150 of the effector genes. And it's not only that it's down-regulating these, at the same time, there's another group of about more than 60 effectors which are specifically upregulated. So this places this transcription factor at a really very prominent node that determines late regulation of the fungus. So I find it amazing that you downregulate basically almost half of the effectors. And among these effectors are some of our favorites. They are really crucial for the early stages of the infection. So why does the fungus not need these effectors later on? And why does it need a set of completely different effectors late during development? We have, of course, no idea yet what these late effectors are good for. And I think that part of this mystery and why you can basically dispose of and don't need so many effectors late anymore is that you actually form this mucilage, this matrix, which may protect the fungus from plant defense responses. And I think this is something we can now begin to analyze if we manage to pull out the correct genes for the biosynthesis. So this was a very unexpected finding. I never wanted to work on regulation of effectors. But seeing this, I think to understand the function, we have to keep this in mind, that they are not needed throughout. There's actually a dramatic switch imposed by the fungus, and I hope we figure it out why. Okay, my group. So the people who have been deeply involved in the work were Libera Lopresti. She set up the effector translocation system. Marie Tolo, she did all the work on ROS1, and Shigeyuki Tanaka, who did all the work on the TIN2 effector. I would also like to mention several people who helped us with transgenic plants, and in particular I want to mention Bernd Zechmann at Baylor University for providing us with much needed EM data. And I want to mention two previous co-workers, Armin Jamai and Kerstin Schipper, who contributed to all these unsuccessful attempts. No, they did really a fantastic job, and they also accomplished other things. Okay, this was sort of done on the side, so don't worry, they are doing fine. Okay, and with this, I'm really at the end, but and people who know me know there is always some pictures which I'm allowed to show, because I have decided to work on this system and no other system. So paradise. Paradise is something you probably don't regularly think about. But in Mexico, in the Aztec culture, paradise is very different from what you may associate with paradise. And I find it fascinating that the way you get there is with a lot of water. But then once you have gone through this experience, which must be pretty traumatic, you end up in an environment which is sort of quiet. People are happy doing something. And if you look closely, they are also growing corn. And it's not just that they are growing corn, but if you look really close, you see, I mean, this looks like a fine flowering corn plant, but this here is clearly an infected. Okay, so if I ever manage to go to paradise, I know my fungus is already there. And the reason why it's there and why it's so famous in Mexico is you can eat it and you can actually buy it on the market. Preparation, there are nicer things, okay? Okay, but you can then cook it. And what comes out is quite delicate. You just have to get around that it's all black. But the taste is really fantastic. And with this, I'm done, and thanks. It was fun.
Time for some questions?
Yeah.
So going back to your first story.
Yeah.
Do you have any idea on a subcellular level where any of the cytoplasmic effectors will compartment? Is it really in the cytoplasm, or do they end up in any other compartments after, say, a pinocytosis or a phagocytosis type of event?
We don't know yet, but it's very clear that one of the cytoplasmic, you mean the cytoplasmic or the apoplastic?
I mean, do they transit
Okay.
through something?
Yeah, no, we don't know yet.
Okay.
So the data I've shown you are pretty new, and we wanted to convince ourselves that the assay is working. Now we will start throwing all sorts of inhibitors on these infected plants. Yeah.
So your transgenic maize plants expressing the VAR-A,
Yeah.
do you know the subcellular localization of the VAR-A protein within the maize plant?
We have not analyzed it. I guess we could, but it should be in the cytosol. I mean, the protein has a MIC tag. We can do the EM, and we better do it, because if you are a referee of our paper,
I think you need And a second follow-up question. There are 300 effectors in
Yeah.
Ustilago. More than likely, some of them are suppressing plant immunity.
Yeah.
Have you taken any of these individual two effectors that you know, or putative effectors, and try to express them in other plants to see if they trigger ETI at all?
this is actually something that is done by Amin Jamai, who is now in Vienna. I have decided not to look in heterologous systems.
But even in maize, and are there wild relatives of maize that would recognize these effectors? And,
We have not.
And within your system, most likely it's being suppressed.
We have tried that very extensively. So far, no. Unfortunately. Yep.
So in Magnum Forte, in the Ascomycete, Barbara Valent sees these interesting modifications of the hyphae and the hyphae
Yeah.
go through plasmodesmin.
Yeah.
Do you see anything like that in Smut?
No. Zero. Zero. So, I mean, all I can say is, I mean, we basically, this idea with hooking up nuclear localization sequence, this is what Barbara has done to actually see the effector accumulate in the big structure and later on finding it in the nucleus. So, honestly, we still are not 100% sure why fluorescently tagged proteins, why we don't see it. So it could be that uptake requires unfolding and refolding does not occur properly. Or it could be that we know if we make a cherry chorus-mate mutase fusion, there's a lot of degradation. So this means the effector that is taken up may no longer have the tag. And we have no idea how much effector is actually being translocated. So our hope was that by forcing it to the nucleus and by using tissue where you basically have no background, that this would allow us to see it. But so far we didn't manage. And I have to say, so I didn't have the time to talk about these other assays. Some of them may not have worked because we always thought cherry is our negative control. Okay? Now, seeing that cherry is taken up, this is not a negative control. So, yeah.
Sorry to get back to John's question. So, what do you know about cell-to-cell transfer of hyphae? I know they get into the xylem, and then I assume that it's easy, but do they go through plasmodesmata when they move from one cell to another, or do you know anything about that process?
We don't know. I'm still waiting to get a maze line that has a plasmodesmata marker that would allow us to visualize that. But it appears, but this is hand waving. Okay, so it appears that when Ustilago goes from one cell to the next, it almost looks as if it's making again an aprosorium. So I'm not convinced that it has to be plasmodesmata. Yep.
Did you repeat the biotinylation experiments on tin-2 with the new conditions to prevent biotinylation during lysis?
Yes.
And it still had the same band?
Yes.
Okay. So that was like the faintest band,
Yeah.
I think, of all.
Yeah.
And also, I was curious, do you have any idea of why bombardment only worked with infected tissue?
Well, what we believe is that the infected, that Ustilago really reprograms the metabolism. And you may have to produce certain amounts of precumarate, which then serves as a precursor for lignification or anthocyanin. And in uninfected tissue, there may simply not be enough precursor.
So maydis has this localized infection,
Yeah.
right? But some of the other Ustilagos don't remain localized,
Yeah.
right? They're systemic and they spread. Is there anything known about what limits the movement of Ustilago? Why does it only go so far and then quit?
To be honest, I don't have any idea. I would love to know. We actually made hybrids between Ustilago maydis and Sporogenesis morellianum quite a while ago. But the analysis got very difficult because we had to make solopathogenic hybrids. And the solopathogenic strains that if they don't need a mating partner, they are not nearly as virulent as when you have a compatible mating. So it didn't help us to address that issue yet.
So we have the endowed tail and white reception for Regina afterwards right now in 3:38. So I hope you'll join us, and also join me in thanking Regina for a lovely